Cancer immunity cycle. How does our immune system recognize and kill cancer cells? This complex yet important question can be answered by using a simple model developed by Daniel Chen and Ira Melman in 2013, termed the cancer immunity cycle. This model involves 7 steps. The cancer immunity cycle starts with a condition in which cancer cells are present. When these cancer cells die, they may release their content. This is the first step in the cancer immunity cycle, the release of cancer cell antigens into the surrounding microenvironment. In the second step, released antigens are taken up by antigen-presenting cells such as dendritic cells. Like other antigens, dendritic cells process the antigens and present them on their surface. The dendritic cells then migrate to nearby lymphatic organs such as the lymph nodes, where they present the antigens to T cells. T cells also wander through the lymph nodes. If a cytotoxic T cell passes an antigen-presenting dendritic cell that has the correct T cell receptor for antigen recognition, this results in their binding to one another. The outcome of this interaction is crucial. The question asked in step 3 is, will the T cell be activated? The interaction between a T cell and a dendritic cell is guided by several positive and negative factors. The most important factor in modulating activation is the connection between the T cell receptor and the antigen, which is presented by an MHC class II molecule. This induces a positive signal inside the T cell, thereby activating it. An example of a negative modulator is the binding of CD80 or CD86 to CTLA4. The connection between these creates a negative signal inside the T cell, inhibiting activation. In simple terms, if the positive signals exceed the negative signals, the T cell is activated and it starts to multiply. In the 4th step of the cancer immunity cycle, these daughter T cells start to patrol the body. They eventually reach the organ containing the tumor. This encounter is facilitated by cytokines directing the T cells, especially if inflammation is present in the affected tissue. In the 5th step, activated cytotoxic T cells infiltrate the tumor. With their receptors, the T cells recognize the antigens expressed on the surface of cancer cells and bind to them. This recognition of cancer cells is the 6th step in the cancer immunity cycle. The 7th step involves interaction between the cancer cell and the T cell. It's the second crucial step in the immune response because it ideally results in the killing of cancer cells and signifies the completion of a single round of the cancer immunity cycle. More specifically, upon binding, cytotoxic T cells aim to induce apoptosis in cancer cells. The interaction is again regulated by a balance of positive and negative signals in the T cell. The most important positive signal is the interaction between the T cell receptor and the antigen bound to MHC class I on the cancer cell. A negative signal is the link between the PD-1 receptor and its ligand PD-L1. The negative signal can prevent T cell-mediated killing of cancer cells, which would leave the cancer immunity cycle uncompleted.